Hello and welcome everybody to my channel ominibioentrance, and today we will be discussing CSIR PYQs for the year 2015. I will be discussing the applied biology and methodology section. If you are new to my channel, please do like, share and subscribe. So coming to the year June 2015. First question. In transgenic mice, the orientation and location of LOXP sites determine whether Cre recombinase induces a deletion, an inversion or a chromosomal translocation. If a researcher wants to put the LOXP sites in such a manner that only inversion take place, which one of the following construct best justifies their intention? Gene X is the target gene. So I have already told you that if the LOXP sites are on the same DNA strand but are in opposite orientation, then it results in an inversion. If they are in the same direction, if the LOXP sites are in the same direction, then it results in a deletion. So here gene X, in the first option, gene X is not flanked by the LOXP site, so that is not possible. In the next one, the LOXP sites are in the opposite direction, and in third one, the LOXP sites are in the same direction. So the answer here is option 3. The next one. The following genes have been genetically engineered to develop hybrid resistance in plants. Resistance to in which of the above cases the mechanism is based on abolition of herbicide binding to the enzyme? So resistant to glyphosate using the 5-enol pyruvyl shikmate 3-phosphate synthase gene. That is correct. Then Biolophos resistance using the bar gene. That is not correct. Sulfonyl urea resistance using acetolactate synthase gene. So both A and C are correct. Answer is option 4, A and C. Then next one. Somatic embryogenesis is an important exercise in micropropagation and genetic engineering of plants. The following steps are considered as critical for achieving somatic embryogenesis. So what are the steps that are critical for achieving somatic embryogenesis? That is the question, and which of the following combination is correct? Reducing the concentration of sucrose in the medium to half. That is not correct. Addition of hormone 2,4-D, 2,4-dichlorophenoxyacetic acid to induce somatic embryos. That is correct. 2,4-D induces somatic embryos. Reduce agar concentration to 0.6% weight by volume. That is not correct. Use maltose in place of sucrose as carbon source. That is also correct. So both B and D are correct, and answer is option 2. Then next one. A gene from genomic library is screened using hybridization technique. After hybridizing the probe, usually a stringent washing step is given. The following statements are given to explain the stringent washing step. So they have screened a gene from genomic library using hybridization technique. So they want to know what are the statements that explain stringent washing step. Stringent washing takes care of removing unincorporated and non-specifically hybridized probe molecules. That is correct. That is what stringent washing does. Stringent washing is done in solution having high salt concentration and lower temperature. It is not high salt concentration. It is low salt concentration and higher temperature. Then stringent washing is done in solution having low salt concentration keeping higher temperature. That is correct. Salt present in washing solution supports hybrids to stay intact by shielding the interference of water molecules. That is also correct. Salt reacts with the DNA molecule and allows easy dissociation of hybrids. That is not correct. If that was the case, you would not get a signal after hybridization with probe. Then next one, stability of hybrid is directly proportional to temperature. That is also not correct. So answer is option 2, A, C and D. The next one, this is a gene knockout experiment. So before coming to the question, I will tell you something about gene knockout. So what is gene knockout? In this, a gene of interest is removed and in place of that another gene is placed. Usually a marker gene is placed. So the gene of interest is replaced with an engineered DNA construct in which the original gene is disrupted by neomycin resistance gene. That is called as gene knockout. So in place of the original gene, another gene is, which is a marker gene, is usually placed. And this here the question is usually based on homologous recombination. There are other ways also to do knockout of the gene. But here the question is based on homologous recombination. So these are the steps which are involved in homologous recombination. So this is the targeting vector. Targeting vector, it has got a neomycin resistance gene and a TK. TK is the thymidine kinase gene, and the TK is present means it is ganciclovir sensitive, ganciclovir sensitive gene. It is sensitive to ganciclovir. This is the targeting vector. Then it is introduced via electroporation to embryonic stem cells. Then you will get recombinants as well as non-recombinants. So if it is recombinant, the targeting vector combines with the target gene. The TG is the target gene. This is the targeting vector. It combines with the target gene at using homologous recombination at homologous sites. That is, these are the homologous sites. These blue ones, these are the which are flanking the neomycin resistance gene. These are the homologous sites. This is the target gene. These are the homologous sites of the target gene. So they will undergo homologous recombination at these sites, and in place of the target gene you will get the neomycin resistance gene. TK will not be present in the target gene after recombination. So what will be the result of the knockout? You will get a gene which is neomycin resistant as well as ganciclovir resistant, because TK is not present, so it will be ganciclovir resistant. Then in the next condition, if it is a non-recombinant, if it is a undergoing homologous recombination at random sites in random chromosome. This is not our target chromosome. It is the random chromosome. It undergoes homologous recombination. So in place of that, you will get the TK gene as well as the neomycin resistance gene inserted in the random chromosome. So the result will be neomycin resistance and ganciclovir sensitive. They are your non-recombinants. So that is how homologous recombination is done for constructing gene knockout experiments. So based on this question, based on this figure, they have asked questions for some 2-3 years, I think. I don't remember exactly, but for 2-3 years they have asked this same question in different ways based on this principle of homologous recombination. So remember this picture. I won't be explaining it again and again. The next one. A student, while constructing knockout mice, isolated mouse embryonic stem cells and introduced an engineered DNA into the cells. However, none of the mice were transgenic. On checking the cells containing DNA construct, he found that he had made a mistake in constructing the DNA, since the cells were resistant to ganciclovir but sensitive to G418. G418 is neomycin resistance. Which of the following construct had he designed? So usually if it should have been transgenic, you should have got only neomycin resistance gene but ganciclovir resistance because TK gene is not involved. But what did he get? He got cells which were resistant to ganciclovir. That means in place of neomycin, TK got introduced. So which of the following constructs had he designed? So how was his construct designed? That is the question. So if we see the first condition, it is your normal. These are the sites of homology. This is the neomycin resistance flanking the sites of homology. This is the TK. This is the normal construct. So he should have got a knockout mice if he had used this particular vector construct. So this is not correct. Here neomycin resistance gene is outside of the gene of interest, and TK gene is inside, but you will not get resistant to ganciclovir. Cells were resistant to ganciclovir. If this construct was used, then in place of here in this figure, in place of neomycin resistance, you would have got a TK over here, and it would have resulted in ganciclovir sensitive. But he got resistance to ganciclovir, so this construct cannot be used. If you see the third construct, neomycin resistance gene is outside of this particular gene of interest. TK is also outside of this particular gene of interest. So naturally you will get a transgenic mice which is ganciclovir resistant as well as sensitive to G418. So option 3 is the answer. The next one. In response to a drug, changes in protein levels were examined in a cell line. A pulse chase experiment was performed using S35 labeled methionine. In comparison to untreated sample, the following observations were made. Few minutes after simulation, protein X accumulates, and this was followed by reduction in protein Y and Z. So here in this experiment, a pulse chase experiment was performed using S35 labeled methionine, and after a few minutes of simulation, what did you observe? Protein X accumulates, and its accumulation was followed by reduction in protein Y and Z. The correct interpretation of these observations would be, so protein X is a protease which degrades Y and Z. That could be the reason, but they have not mentioned anything regarding protein X being a protease, or protein X is having some protease like action or something like that. They have not mentioned. They have also not mentioned what kind of protein are Y and Z. Then protein X is a transcriptional repressor that controls expression of Y and Z. That also could have happened, but we don't know. They have not given full details regarding whether it is a transcriptional repressor, whether it is a protease, etc. They have not mentioned. Expression of protein X and loss of Y and Z are unrelated. That also could be the reason. But we are not sure because information is very less in the given question. So answer is option 4. Information is not sufficient to distinguish between the three possibilities stated above. Then next one. A protein undergoes post-translational modification. In an experiment to identify the nature of modification, following experimental results were obtained. Protein moved more slowly in an SDS page. That means it has undergone some kind of post-translational modification. That is why it moves slowly in the SDS page. Now, isoelectric focusing showed that there was no change in the PI. That itself means that it is either glycosylated. Mass spectrophotometric analysis showed that the modification was on serine. The modification that the protein undergoes is likely to be glycosylation. That is why there was no change in the PI value. So answer is option 2. Coming to the next year, December 2015. A gene expressing a 50 kDa protein from an eukaryote was cloned in an E. coli plasmid. under the lag promoter and operator. Upon addition of IPTG, the 50 kDa protein was not detected. Which of the following explains the above observation? So here a gene expressing 50 kDa, a gene which produces a 50 kDa protein was cloned in an E. coli plasmid under the control of a lag promoter and an operator, and IPTG was added to it so that you will get a sorry. Okay, upon addition of IPTG, the 50 kDa protein was not detected. So you added IPTG to induce the lag promoter and operator, and the 50 kDa protein was not detected. Then, which one of the following explains the observation? Here, it is called as this problem is usually encountered in researches, and this problem is called as codon bias or differences in codon preference. So, different organisms have different preferences for the codons that encode specific amino acids, and all this is evolutionarily controlled. Suppose you know that an example I can suggest. It is only just an example for you to understand. You have 4 codons for alanine, and I will say only about regarding 2 of the codons. So E. coli prefers the GCA and humans prefers the GCU codon. Okay, so E. coli prefers GCA and humans use GCU codon for alanine. And when you try to express a human protein in E. coli will fail to recognize the GCU as a codon for alanine, and it because it has preference for GCA than GCU. So the functional protein will not be formed. This is called as codon bias or differences in codon preference. So answer here is option 3. Then next one. Neomycin phosphotransferase gene frequently used as a selection marker during plant transformation inactivates which one of the following antibiotics? So it inactivates kanamycin. Answer is option 4. Then, which of the following techniques will you use to identify more than 1000 differentially expressed genes in normal and tumor tissues in one single experiment? So in one single experiment, you should identify the differential expression of the gene. So that can be done using transcriptome analysis. The next one. For identification of 3 proteins moving together as a single band upon loading in a single lane of an SDS page gel, the best method is. So here the proteins, there are 3 proteins and all the 3 are moving at the same molecular weight, or I can say that they are moving together as a single band, and upon loading on a single lane of SDS gel, how will you identify the 3 different proteins? So that can be done by using Western blot followed by stripping and reprobing. This I have explained in one of my previous year questions, and I have told that time also that this question has been repeatedly asked. So first they have asked for a part C as a part C question, and now they have asked it as a part B question. So you can refer to that question which I have discussed earlier. So answer is option 3. Then next one. 4 single amino acid mutants A to D. 1 minute. So these are the 4 single amino acid mutants A to D of a protein in the epitope region of a monoclonal antibody X were made and expressed in E. coli. The lysates from the 4 E. coli cultures expressing these 4 proteins were run on an SDS page gel, subsequently transferred to nitrocellulose membrane and Western blotted using monoclonal antibody X raised against the wild type protein. The results are presented in the figure below. So 4 single amino acid mutants A to D, these are the mutants A, B, C and D of a protein in the epitope region of a monoclonal antibody X. And what is this epitope region? It is where your antibody will bind, where your antibody will bind. So only then you will get a signal in your Western blot. Okay. So at the region where the antibody binds, they have made single amino acid mutants and expressed in E. coli. The lysates from the 4 E. coli cultures. So they isolated the protein from these 4 E. coli cultures and they ran an SDS page gel and they transferred it to a nitrocellulose membrane and they conducted a Western blot experiment using monoclonal antibody X raised against the wild type protein. So they expect that this antibody should go and bind to this particular epitope region which they have mutated. The 4 single mutations upon sequencing were found to be valine to alanine, glycine to proline, alanine to aspartic acid and isoleucine to leucine. So before coming to the answer, I would like you to know what is called as conservative replacement. In this particular type of replacement, an amino acid in a protein is replaced by a different amino acid but with different biochemical properties. That is, it will have same charge, hydrophobicity, size, but it will be an entirely different amino acid. But it will replace the first amino acid by a second amino acid in such a way that second amino acid is having the same biochemical properties as your first amino acid. And proline is the least preferred for such kind of substitution experiments. There is no amino acid that could replace a proline. Okay, now coming to the question. Now B is due to V to A, that is valine to alanine. B, you got a band over here, valine to alanine. Valine and alanine have almost the same biochemical properties. So you will get a band upon binding the binding of the monoclonal antibody X to the mutated epitope region where valine has been replaced by alanine. So you will get a band over there. And C is due to G to P, glycine to proline. I told you already, proline cannot be replaced by any other amino acid. There are no partners for proline. So glycine to proline replacement cannot take place. So C is also correct. Sorry, option 1 is also correct. That is, B is due to V to A and C is due to G to P. 1 is correct. We will see the other options also. Then you will understand what I meant. B is due to G to P, glycine to proline, but you got a band over there. So it cannot be G to P. And D is due to V to A. If it was valine to alanine replacement, then D would have given a band, but that is not the case. Then D is due to isoleucine to leucine. If it was isoleucine to leucine, then they both are similar biochemical properties and you should have got a band, but you didn't get a band in D. And A is due to A to D. A is due to A to D. What is A? Alanine to aspartic acid. So alanine and aspartic acid, they have got dissimilar biochemical properties. One is an acid, it is negatively charged, whereas alanine is not acidic, it is neutral. So you should not have got a band, but you got a band over there stating that option 3 is also not correct. C is due to V to A. V to A means you should have got a band, and A is due to I to L. Isoleucine to leucine. But you got a band over A. That might be the correct answer. But V to A replacement is not correct, because if it was valine to alanine, then C should have given a band, but there's no band over there. So option 4 is also not correct. Then next one. A hypothetical operon involved in the synthesis of an amino acid X is on in the presence of low levels of X. So when the concentration of X is less, the operon is on, and off in presence of high level of X. So when there is high level of X, the operon is off. The symbols A, B, C in the table below represent a structural gene for the synthesis of X synthase, the operator region, and the gene encoding the repressor, but not necessarily in that order. Okay, so A, B, C, they represent the synthase gene, the repressor, and the operator, but it is not in the correct order. From the following data, in which superscripts denote wild type or defective genotype, identify which are the genes for X synthase, operator region, and the repressor. So we have to identify the genes. So if you see strain 1, A minus B plus C plus, and X synthase activity is detected, that means A doesn't code for X synthase. It can be either B or C that is involved in the synthesis of, that is involved in X synthase as as X synthase, because at low level of X it is detected, and high level of X synthase activity is detected. Then next one, C minus, A plus B plus and C minus, it is detected and also detected at high level of X. But B plus and X synthase activity is detected, so B can be the one coding for X synthase. Then if you see the third one, A plus B minus C minus, not detected and also not detected at high level of X. So activity not detected, it is B minus. So even at high level or low level, activity is not detected because it is B minus. So confirm B as X synthase only. B is coding for X synthase activity. X synthase enzyme. Then next one is a mirodiploid. A plus B plus C plus, A minus B minus C minus. Detected at low levels but not detected at high levels. So high level of X, the repressor binds to the operator and prevents the transcription and translation process. Because they have already told that at high level of X, the operon is off. That means the repressor is active and it will bind to the operator region, and it will result in no X synthase activity. Then next one, strain 5, A plus B plus C minus, A minus B minus C plus, detected and not detected at high levels. So repressor is transacting. If C is the repressor, then at higher levels of X, no activity will be detected. So C could be the repressor as per strain 5. Then strain 6, if you see A minus B plus C plus, A plus B minus C minus, activity is detected at low and high levels. So A is the operator. A is mutated, so C cannot go and bind, and there and X activity will be detected both at high and low levels of X. So the order will be B, A, C for X synthase. A is the operator and C is the repressor. Answer will be option 4. Then next one. A protein has 4 equally spaced trypsin sensitive sites. So this is the protein. It has got 4 equally spaced trypsin sensitive sites. 1, 2, 3 and 4. Which results in peptide fragments. This is 1, A1, A2, A3, A4 and A5 upon digestion with trypsin. The peptides A2 and A5 represent N terminal and C terminal. So you have got A2 over here and A5 over here. This is your N terminus and this is your C terminus. What I have drawn here, A1, A2, A3, A4, A5, that is not the order in which the fragments are present. So they have given in the question A2 is at the N terminus and A5 is at the C terminus. Sorry. Now you are asked to synthesize this protein. At time t is equal to 0, you added all the 20 amino acids labeled with C14 and initiated the synthesis. So protein synthesis, as you all know, it starts from N terminal end to C terminal end. At time t is equal to 4, full length protein is synthesized. So it requires only time t is equal to 4 minutes or hours for full length synthesis of the protein. If you stop the synthesis of the protein in time t is equal to 1 and digest the protein with trypsin, which peptide will have maximum C14 label than others? So at the full length synthesis, it requires 4 minutes or 4 hours or 4 seconds. Full length synthesis require 4. So now they have asked for, now if you stop the synthesis of the protein at time t1, so at time t1, I told you already, protein synthesis starts from N terminal end to C terminal end. At time t1, the N terminal end has already been synthesized. That is sure for 100% guarantee. And digest the protein with trypsin. Then you digested the protein with trypsin. Which peptides will have maximum C14 label than others? They have asked which peptide will have maximum C14 label. Maximum C14 label, I told you that it will be the A21 because it is at the N terminal end. Protein synthesis is from N terminus to C terminus. So naturally A2 will be having maximum C14 label than others. So answer will be option 4. So next question. In an organism, expression of gene X is induced in the presence of a hormone. Genetic analysis showed that the hormonal signal is transduced through two proteins, LET1 and LET2. The expression of gene X was studied in lines overexpressing the active LET proteins, knockout of the LET proteins, or a combination of both. Results of the expression of the gene in the presence of the hormone is summarized below. So LET1 is overexpressed, means expression of gene X is there. LET2 is overexpressed, expression of gene X is there. If LET1 is knocked out, no gene expression. If LET2 is knocked out, no gene expression. LET1 is overexpressed and LET2 is knocked out, no gene expression. If LET1 is knocked out and LET2 is overexpressed, there is gene expression. So based on the above, which of the following pathways best fit the observation made? So if you see the first pathway, I will read it out to you. It is not clear. This is hormone. This is LET1, LET2. Hormone activates both LET1 and LET2. LET1 and LET2 come in combination will cause expression of gene X. So if you see the first pathway, it is not correct because if the hormone activates LET1 and LET2, then knockout of any one of these will not lead to the expression of gene X. But this is not the case, as you can see in the tabular column. So LET1 is overexpressed and LET2 is knocked out, means there is no gene expression. But if LET1 is knocked out, and LET2 is overexpressed, there is expression of the gene. But actually you should not have got expression of the gene in the second case. But if it was as per the first pathway. Now if you see the second pathway, hormone activates LET1, it activates LET2 and LET2 activates gene X. So if LET1 is overexpressed and LET2 is knocked out, the gene will not be expressed because let2 is not present. But if let1 is knocked out and let2 is overexpressed, the gene will be expressed by let2. So the option 2 is the right pathway. Then next one. A mixture of two proteins was subjected to following three chromatographic columns: A cation exchange, B size exclusion, FedEx 100 and reverse phase. So you all know what are these principle behind cation exchanger, size exclusion and reverse phase. I am not going into detail about all those things. They have been studied from your BSc level onwards. Following elution profiles were obtained. So this is cation exchanger, this is size exclusion and this is reverse phase. Which of the following statement is correct? So here if you see the cation exchanger, the anions which are more anionic, they will bind to the cation exchange column, and the more anionic it is, it will be eluted last. So B is more anionic as per this particular graph. B is more anionic, that is why it eluted in the end. Size exclusion, the A is smaller, that is why it got eluted last, and B is larger, so it eluted first. So A will enter through the pores of the FedEx and it will elute last because of its smaller size. Then in reverse phase chromatographic column, A is more hydrophobic than B. So that is why A got eluted in the end. Now we will see what they have written, the statements. A is larger and more hydrophobic than B. A is not larger as per size exclusion chromatography. A is smaller. So option 1 is not correct. B is more anionic. Okay. And more hydrophobic. B is not more hydrophobic. A is more hydrophobic. That is why it got eluted last. So 2 is also not correct. A is more hydrophobic. A is more hydrophobic. Okay, that's correct. And smaller than B. Okay, here as per size exclusion, you can see A is smaller than B. So option 3 is correct. A is more cationic and smaller than B. That is also not correct. So option 3 is the correct answer. Then next one. The following statements are related to plant tissue culture. Friable callus provides the inoculum to form cell suspension cultures. That is correct. The process known as habituation refers to the property of callus losing the requirement of auxin and cytokinin during long-term culture. Sorry, that is also correct. Cellulase and pectinase enzyme are usually used for generating protoplast cultures. That is also correct. During somatic embryo development, torpedo stage embryo is formed before heart-shaped embryo. It is globular, heart-shaped and then torpedo stage. Not torpedo followed by heart. It is heart followed by torpedo. So D is not correct. So A, B, C are correct. Answer is option 1. Then next one. A researcher is studying the subcellular localization of a particular protein X in an animal cell. So he wants to know where is this particular subcellular localization, where this particular protein X is going to be located in the animal cell. The researcher performs successive centrifugation at increasing rotor speed. Centrifugation at increasing rotor speed. The researcher starts spinning the cellular homogenate at 600 G for 10 minutes. Collects the pellet, spins the supernatant at 10,000 G for 20 minutes. Collect the pellet, spins the supernatant at 100,000 G for 1 hour. Collects both pellet and the final supernatant. On subjecting various pellets and final supernatant to Western blotting with anti-protein X antibody, the protein X is observed to be maximally expressed in pellet after centrifugation at 10,000 G. Based on the above observation, what will be the most likely localization of protein X? Now, if you remember this, you can easily answer this particular question. The question is really very big, but it is very simple. Now the researcher, he want to study subcellular localization of protein X in the animal cell. So what does he do? He performs a centrifugation experiment by increasing the rotor speed. So first he will grind the cell and will make a homogeneous preparation out of that called the cellular homogenate and spin at 600 G for 10 minutes, collect the pellet, then again he will collect the pellet, spin the then take the supernatant and spin it at 10,000 G for 20 minutes. Again he will collect the pellet, spin the supernatant at 100,000 G for 1 hour, collect both pellet and final supernatant. That is the process I have written over here. Filtered homogenate, and if you spin at 600 G for 10 minutes, nuclei will be separated. If it is at 10,000 G or 15,000 G, mitochondria, chloroplast, lysosome and peroxisomes will get separated. If it is at 100,000 G for 60 minutes, microsomes will get separated. And if it is at 300,000 G for 2 hours, ribosomal subunits and polyribosome get separated. Now, on subjecting the various pellets and final supernatant to Western blotting with anti-protein X antibody. So, he ran a page gel, then transferred it to nitrocellulose membrane and treated it with anti-protein X antibody. The protein X was observed to be maximally expressed in pellet after centrifugation at 10,000 G. So at this second centrifugation step, the protein X was observed to be maximally expressed. So based on the above observation, what will be the most likely localization of protein X? So it is at the second step, means it is mitochondria that is getting separated. They have already told it is animal cells, so chloroplast cannot be taken into consideration. So it is mitochondria that is the protein X is located. So answer is option 3. Then next one. You have transiently expressed a new protein for which no antibody is available in a cell line to establish structure-function relationship. Which one of the following strategies is the most straightforward way to examine the expression profile of this new protein? They have asked for the most straightforward way to examine the expression profile of this particular protein. So we will see the options. By metabolic labeling with S35 labeled amino acid. That is not a straightforward way. Then making a GFP fusion protein with this new protein. That is very easy. By using fusion vectors or expression vectors, you can make a GFP fusion protein with this new protein. The GFP anti-GFP antibody could be used because for the new protein, antibody is not available. So anti-GFP antibody could be used and Western blotting could be done. So that will be one of the straightforward way to examine the expression profile of this new protein. Then immunoprecipitating this protein with the help of another protein for which antibody is available. That is not correct. Running SDS page and identify the protein. That also is not correct. Just by simply running SDS page and then identify the protein, that is not correct. So answer is option 2. Then next one. During an experiment, a student found increased activity of a protein for which there were 3 possible explanations. Increased expression of the protein, increased phosphorylation, or increased interaction with other effector proteins. After conducting several experiments, the student concluded that increased activity was due to increased phosphorylation. Which of the following experiment will not support or provide the correct explanation drawn by the student? Western blot analysis can provide Analysis of transcription rate. They have already told that after conducting several experiments, the student concluded that increased activity was due to increased phosphorylation, and phosphorylation is a post-translational modification. So analysis of transcription rate will not provide correct explanation. So answer is option 2.